So I'm going to show you how to use the differential gene expression tool inside of NeMO Analytics to create a gene list that is either up or down regulated genes of a condition that's interesting to you that you can then save as a gene signature that you want to go explore in other data sets. So here's the front page of the current version of our manuscript. And this link here will take you to NeMO Analytics, the landing page for the neocortical development collection, which this paper is all about. And that's here. This will bring you here. And this just gives you an abstract and kind of a layout of what's going on. We can look at individual gene links for all of the different data collections. We're interested in the adult neocortex layers for this particular analysis. So we're going to click on this. And that's going to bring us to this page, the adult cortical layers. I already have a gene loaded here that has some layer specificity to it. But the reason I'm coming here is to show you the next thing. So every data set in NeMO Analytics has this little 3 dots next to it. You can look at different ways to display the data, some information about it. And one of the other things you can do is the comparison tool, which is a method of differential gene expression within that data set. So if we click on that, that will bring us here. So it's preloaded with the data set we were looking at, which is right here. This is the Ma et al. DLPFC study across humans and primates. And what we're going to do, we're actually going to look at the subclasses of cells they have listed here. We're going to look at expression of genes in a couple of these layer 3 to 5 IT groups, the intratelencephalic projection neurons that they have 3 classes of. And in the paper, we show that one of these classes is primate specific, specifically this -2 group of neurons. So here, what we're going to say is we want to look at the subclass of cells, and we want to compare that layer 2 class. We're just going to compare it to the other 2 classes that are very close to it. So these are going to be genes that are distinguishing between subclasses of this layer 3 to 5 intratelencephalic neuronal subtype that they found, specifically the 2 versus the 1 and the 3. So then we could filter data and look at other things. We don't really want to do that. I don't want to give it a p-value or a full change cutoff. I just want to let it plot these for us. So if I hit plot now, it's going to plot the average expression of genes in that 2 class versus the average expression in the 1 and 3 class. So this might take a second. It's a relatively large data set. While that's working, though, I'll let you know a little bit where we're going. So again, this was the subtype of neurons that is apparently primate specific. And we're now trying to capture a gene signature for it by contrasting it to closely related neuronal subtypes. And then we'll have a gene list, not only that is interesting to look at it in itself, but it's interesting to look then at the expression of that gene list that's coming from this primate-specific neuronal type in other data sets. See where else it's expressed and where else it is. Okay, so now it's come up here now. So then I can use tools here to grab genes. I could have used those p-value cutoffs and full change cutoffs to select genes. I kind of like to get a lay of the land here and look at these genes first. But the idea is, however systematically you do it, is to grab genes that you're interested in. So here, these would be genes that are upregulated in the -2 neuronal type. Oh, look at that. Hold on for a second. It's waking up for us. So this is just showing you can grab them, or you can be systematic about those cutoffs we were looking at. Going like this, we could just grab these genes here that are upregulated in the -2 subtype versus the other two. They list them right here for us, along with their full change. So here's FOXP2 that was mentioned in the report as one of the genes in this primate-specific neuronal identity. So now we can go down here and say save only the selected genes, and that'll save them as unweighted. It'll just save the list of this genes that we can then come back to. And then we'll call this Ma et al. 2022 layer 3 to 5 IT-2 genes up. So upregulated in this subtype. And we'll say here, hit the save button. Okay, so now that's saved. If I go over here now to hit this gene list button, there it is. We just created this. It's private now. We could set that to be public. But that means that because this gene list is now here, we can look at this list of 25 genes. Here they are. That FOXP2 should be on there. Yeah, that we just saw. So these are those 25 genes we just saved. We now click on the projection tool. The neocortical layers here is the data collection we want to look at. This has a lot of the single nucleus RNA sequencing from layer-specific neurons. And the pattern we want to look at is from the Ma et al. That we just I've made a couple that sound about the same. This is the one we just made and I named up. So I can click on that one and hit proceed. I hit this now, and it's going to project that list of 25 genes into all the data sets in this collection. So every image, instead of being an image of the expression of a single gene in one of those adult layer data sets, it will be the expression, the composite or summed expression of the 25 genes on this list. So the first time we do it, it takes a minute because it's never seen this gene list before. So that's why it's saying it's going to take a few minutes. And as you can see, there's lots of data sets in this collection. But now after we do this, once it comes up, it will never take that long to load again because it saves that pre-calculated projection in relation to this set. So now, shortly, they should start popping up. So what this does is actually, for each data set, it spawns a separate job and goes away, looks up the 25 genes for that every cell in that data set, sums them up, and then returns them. And like I said, actually saves them for later use as well. So now it looks like it's loading the display. At least for some of these, it's completed. The projection is coming back. Again, this first time looking will be the longest. It won't ever take this long again. And now we'll be able to see, as these data sets start to come in, the expression of our 25 genes that are specific to this neuronal type, at least specific when compared to the other neuronal types that are closest to it. And so this sort of kind of analysis is as versatile as you can think up different things to either do differential expression on in NeMO, as we just did, or you can also, if there's an interesting list of genes in your own work, or a paper online, or a supplemental table, you can upload that list of genes to NeMO and project it to see that. So, and here the algorithm for this is not too important. When you project a principal component, the math of it actually boils down to summing up the weighted expression of each gene in the principal component. And that works fine for us here. These 25 genes, they don't have weights. When there's no weights, it just assumes each one is 1, and all 25 genes are summed up. There's lots of different ways to project here. Just counting how many of these 25 genes are expressed in any of your cells, or if you had a PCA or NMF algorithm output a whole dimension, we also have options for that. Okay, still waiting for these to be displayed. If we can. I think they're created now. I'm just got to get it to. Great. So it looks like these are starting to load now. As I said, that took a long time the first time, but in the future, if we ever load the same projection again, it should be quick. So already we're seeing some kind of interesting thing here, or at least what we expected to see. The expression of this packet of genes is high. Whoops. It's high in the neuronal subset that we chose to grab the gene list from, which is here, this one, the layer 3 to 5, IT-2, and lower in the two that are very close to it that we contrasted it with. The other data sets, this is the Jorstad 2023. This results in identifying a subpopulation of one of their layer 4 identities up here. There's several of them. The layer 4AB and layer 4C are both the darker blues. They're from visual cortex. And it looks like a subset of the layer 4 identities that are not in visual cortex actually correspond to this -2 subtype that we got out of the MA data. And this one is from DLPFC. So you always got to kind of keep in mind where things are coming from. And to see that layer specificity and regional specificity here, we can choose a different display that shows us which regions these came from. So there had been split into 3 donors by layers. We can't see donors here, but we can see the regions they came from, and then the layers again in color. And again, the 2 visual cortex dissections of layer 4 A, B, and C are not very high in this, but outside of V1, the layer 4 identities, this lighter blue, actually show that that's what's really high in the IT-2 signature data set. Again, here, it looks like it's somatosensory cortex, auditory cortex, temporal gyrus. All these outside of V1 layer 4 identities share that. So, and this is just now like any other page in NeMO, except we're looking at a very specific gene expression signature. So again, in the MOZ study, they had human, chimp, macaque, and marmoset, and in each one of these, we see the specificity of the contrast we just did. In other data sets, as I showed you in the Jorstad, this is another human study in temporal gyrus from Hodge et al. A few years ago. We zoom in on this one, you can't really see because it's squished, but you can see here enriched in layer 4 this identity. And again, this was supposed to be the identity in Ma et al. That was connected to the primate-specific expression. So this is a mouse study where it really, layer 4 is here in blue. It does not look like it's popping up above everybody else, but it does look like this isn't perfectly absent in the mouse. If we look at this study now, there is something of an enrichment in layer 4. It's not like it is in the human study. So there might be some overlapping here, and we could dig down into the individual identity of each of those 25 genes and look at them either in a heat map that we can design here in NeMO or one by one on a display like this. The spatial data show it's this layer specificity of this packet of genes that we already knew about. It's also very nicely visible here in the macaque adult spatial data. Just zooming in on this so we can see, you can see this little ribbon of layer 4 lighting up in the spatial transcriptomics. If we look then though at the mouse spatial transcriptomics, we don't see the layer 4 signature. And this is really where the observation that this is likely to be primate specific came from. Not this data in particular, but when the Ma et al. Study looked at non-primate mammals, they didn't see it either. So there's nothing here in the mouse, but this newer study that has a little bit more high resolution expression data, also spatial transcriptomics, shows us that there may be some very specific pieces, and this looks like perhaps cingulate here. There may be some cortical regions in the mouse where it actually does appear. So it might be specific to the primate in the regions that particular studies have looked at, but getting together tons of data like this often pops up things like this that are kind of surprises. Clearly, it is not expressed highly in the cortical plate, or excuse me, in this wide section of cortex here in a layer-specific manner. But it looks like cingulate might have some expression. So I just wanted to show you that as an example of what you can do. And again, this is as versatile as it gets. You could do differential expression on any data set in NeMO Analytics to get a gene like this, or you could come up with a gene list from outside and upload it to visualize it across any of the data collection. Hope it was useful.